So this is basically the structure of our synapse. So we've got our presynaptic neuron, so that's the neuron that's before the synapse. You've got your postsynaptic neuron. In the presynaptic neuron, we've got a synaptic knob. Postsynaptic neuron, we've got neurotransmitter receptors that we don't have on the presynaptic one. Right? This is to make sure that your nerve impulse continues to go in the right direction. Right, and in between that, so your synapse, so the proper phrase that we need to start using for the synapse is the synaptic cleft. Right, so the first thing that happens is our action potential, because remember that we're going and we're calling a nerve impulse an action potential. So the action potential goes down and it moves down the nerve cell. Right, remember that that is caused by the depolarization of the neuronal membrane. Right? So as that action potential moves down, it causes the depolarization of the membrane in the synaptic cleft. Right? That depolarization of the membrane in synaptic cleft causes voltage-gated calcium channels to open up. So now, right, remember that your neurons, they're in like tissue fluid, and in your tissue fluid, you've got loads of different ions and stuff, right? So that causes and allows calcium ions to move down the diffusion gradient into your neuron, right? Once they move into the neuron, they go and they bind to these synaptic vesicles, right? Remember, all a vesicle is like a little membrane-bound sac with something in. That causes these synaptic vesicles to go and move towards the edge of your membrane with neurotransmitter, right? I've not called it acetylcholine yet, but it is, right? Where the neurotransmitter then diffuses across the synapse, right? Because it's going from an area of relative high concentration to a relevant area of low concentration. Right, it happens really quickly because your synapse is like Microsoft. There's not much of a gap. Right. So the neurotransmitter diffuses across. Right. As it diffuses across, it then binds to a postsynaptic membrane. So it binds to a neurotransmitter receptor on this postsynaptic membrane. Right, when it binds, right, remember a receptor is just a protein, right, basically, or like a series of proteins that work together. Right, when it binds to that neurotransmitter receptor, that causes a conformational shape in its protein, which causes, right, so it might go from that, and it just squeezes in a bit. So that goes and it stretches the membrane. As it stretches the membrane, you've got a sodium ion channel. And it causes your sodium ion channel to stretch open because the entire membrane is being stretched. Not very much, ever so slightly. As that sodium channel is stretched, it allows sodium to go and diffuse into your postsynaptic neuron. Remember that sodium diffusing into the cell, right? Think back to what we did about action potentials. That causes an action potential to be generated in this postsynaptic neuron, so your nerve impulse goes and carries on moving along. Right? We don't want to leave this neurotransmitter sat in the synapse like forever, or else your postsynaptic membrane will just carry on firing, and you only want it to fire once because you just want one nerve impulse. So this neurotransmitter, right? Actually, what happens is it's broken down by an enzyme, right? It's broken down by an enzyme called acetylcholine esterase, right? Remember neurotransmitters called acetylcholine, so you could probably get away with calling it acetylcholinease, right? So that breaks it down into choline and acetyl, which can then be reuptaken actively by this neurotransmitter pump right? So it's just reuptaken back into your presynaptic neuron, where they recombined back into acetylcholine, right? So you'll find that in the ends of your neurons, you've got loads and loads of mitochondria in your synaptic knob, because this neurotransmitter is being actively uptaken, actively back uptaken into the synaptic knob, where it can be used again, right? So to get recombined into acetylcholine, and it to go back into a synaptic vesicle.